I'm going to appoint someone to serve as the class officer, the one who ensures the discipline, who picks those who need to say something, so that we can have a smooth lesson. So I'm going to ask Kumla to give you some of the rights so that you can be able to help me with this. So that Michael, rather the class monitor is going to help to mute those who are making noise in their background, and also to choose those who have something to say. So I was asked to discuss with us or with you the topic of inheritance. This is a senior 4 second term topic. I don't know if any of you have covered it yet, but even if you haven't, I believe we can still be able to share about this topic and learn something. So before I go any further, I would like to ask what you know about inheritance. What is inheritance? By show of hands. What is inheritance? Yes, Ricky?
I don't know nothing much about inheritance, but I know it's like the passing down of something from one generation to another.
Okay, thank you, Ricky. Mugabe, you have something else to add on?
Yes, inheritance is the sharing of characteristics from parents to offspring.
Thank you, Mugabe. Farren?
Inheritance is the transmission of genes from parent to offspring.
Okay, you are all spot on. Now, you may find in some books, it goes by another name. Some books will call it heredity. Heredity, while other books will call it inheritance. Others will call that topic genetics.
Come back to this one.
So all of these people will be talking about the same thing, regardless of how they choose to name their Okay, in this topic, we have the following expected competence and learning outcomes. You know, whenever we have a new topic, it's important that we know what we are going to be handling so that we can know the scope of what is we have to deal with, so that we don't read things that are too much or read things that are too little, and then we are surprised when the exams come. So this is the competence. So we are expected at the end of this topic to be able to appreciate that the transmission of characteristics happens from parents to their offspring through a process we call heredity. Heredity. So heredity is another term for inheritance. And then how are we going to achieve this competence? These are the following learning outcomes. So for today, we are going to deal with learning outcome 1. We expect to understand the process of cell division and the significance of meiosis. So in today's lesson, we are going to seek to understand this process of cell division and particularly the process of cell division known as meiosis. And we are going to seek to understand why this process is important in living things. I know previously in the topic of growth and development, you've been exposed to another type of cell division known as mitosis. How many of us have come across the process of mitosis under growth and development? By show of hands. Could I see? Okay, Mugabe, I can see. Ruze, Jethro, Kezabu. Seems a good number of you haven't encountered it. Yeah, but that's, there are quite a number. That is S4 term 1 work. And if you have encountered the topic of mitosis, this one should not be a big challenge for you to understand. However, they are not exactly the same, but they are similar in some ways. And today we shall see in what ways they are similar and in what ways they may be different. So once this learning outcome has been concluded, then we shall go to the second learning outcome, which is to understand the concept of inheritance using genetic diagrams. So this one will be done sometime in the future. And then after that, we shall look at the third one, which is understand and explain sex determination in humans. Understand and explain sex determination in humans. And then lastly, understand and explain sex linkage in humans. So these are the 4 learning outcomes that are expected to be covered in this topic. This is a topic that ordinarily we should do in 20 periods. Those are like 10 lessons. But we'll see what much we can do given the time we have at our disposal. So I want us to get down to the first learning outcome. So we know that cells are the basic building blocks of all living things. If you've heard of what we call the cell theory, that all living things are made up of cells. So there's nothing that is living that doesn't have cells. And because every living thing has a cell, you'll find that the cell makes the functional fundamental unit of a living thing. All the 7 life processes, as we know them, can be carried out at cellular level. Can we remind ourselves of these life processes, the 7 life processes that every living thing should be able to exhibit? Yes, Noble?
Living things should be able to breathe.
Breathe. Could we use another word other than breathe? Mugabe?
Respiration.
Yes, good. So every living thing must respire. Thank you, Mugabe. Let's have another person. What are the other 6? Mugabe has mentioned 1. Can we have the other 6? The class monitor, please help me. I'm getting a lot of background noise. Let's have Marik. Marik, we can't hear you. I think Marik has a problem with their network. So, locomotion? Is locomotion carried out by every living thing? locomotion is preserve of only animals. Okay. So what should be the appropriate word?
Movement.
Yes, so all living things move. Locomotion is a special type of movement that happens in animals, whereby animals they move, they are able to change their location. They move the whole body from one place to another. They transfer the whole body from one place to another. But plants are able to move some parts of their body. So those are two. Can we have the other 5 quickly so that we get done with the lesson?
Excrete.
Rosalind. Okay, let's have Mary, Shanik.
Living things are to excrete.
So there's excretion, where they get rid of waste products of metabolism. Please let us not talk unless we are called upon so that we can be organized. Then let me ask Steven to tell me another one. Faizo.
Locomotion.
Locomotion, we said, is not something for every living thing. We said we'd rather use the word movement. Let's mention the ones that haven't been mentioned.
Growth and development.
Let's have Monique Rocket. Monique Rocket.
They respond to stimuli.
Yes, irritability or response to stimuli. Or who else? Let's have Stephen. Stephen. Okay, let's have Emmanuel Sekaita.
Okay, growth.
Yes, they all grow. And let's have ITEL P17.
Reproduce.
Or reproduce. Okay. What has been left out? Vivo, tell us what has been left out. Okay, so there are 7. We usually say Mrs. Green. M for movement, R for reproduction, S for sensitivity, G for growth, R for respiration, E for excretion, and N for nutrition, or F for feeding. Now, all those processes, as we have mentioned, take place at cellular level. The unicellular organisms are able to carry them out in those single cells. All those 7 life processes. But multicellular organisms equally also carry them out. Now, we know that, we know that for organisms to continue to exist, there must be cell division. And what happens here is that a cell divides and forms 2 daughter cells. And why must this happen? One is that if the cell doesn't divide to form new daughter cells, then eventually that cell will die and that organism, it means it will be in that state. It will never increase in size. It will never increase in size. So this is very important. This is very important because it helps organisms to grow. But also once in a while, these organisms experience certain challenges, like they get damaged and their tissues must be repaired. So cell division is equally important for the repair of worn out or broken tissues. So that means the existing cells must divide and form new cells, which will replace the damaged tissue. Like when we get wounds or we get cuts, those wounds will heal because the tissues surrounding those cut surfaces divide and cover up that area. And of course, this is very important because it helps to cause the person to survive infections, because if the wound remains open, then they may get infections, and the infections can cause them to get diseases or eventually die. Or the organism may not even function properly because the damaged tissue is not able to do the work that is expected of it. For instance, if you break a bone, then you may not be able to move your body part because the bone is broken. And if that part is very important, it will affect your life significantly. But we also know that cell division is important for reproduction. And you will know that there are two types of reproduction. What are the two types of reproduction? Who can tell us? What are the two types? Yes, Kezabu?
Sexual and asexual.
Would you mind telling us the difference between the two? Between sexual and asexual reproduction?
In sexual reproduction, gametes are used, while in asexual reproduction, they are
Okay, Stacy, please add on. Nancy, Stacy.
I was going to say that in asexual reproduction, there is no use of gametes. In sexual reproduction, the use of sexual gametes, like
Okay,
the sperm over.
Okay, that's correct. Now, what happens also in sexual reproduction, there are two individuals involved, but for asexual reproduction, only one individual is involved. Only one individual is involved. And so that is why there's no need for gametes, or what we call sex cells. Now, asexual reproduction, as you might already have learned, it takes place by mitosis in the higher organisms, okay? Like a protozoa, like a fungi, like mammals, like animals, plants. When they reproduce asexually, they are carrying out a process called mitosis. But then those that don't reproduce asexually, they must find another type of cell division that enables them to produce sex gametes. And this special type of cell division is what we call meiosis. Yes, Rosalind, your hand is up. You can tell us, you can share with us what you want to say. Oh, she has put her hand down. So today we are going to look at a special type of cell division called meiosis. And since we have previously looked at mitosis, this should not be a challenge for us to understand. But before we go there, let us have a quick recap of what mitosis is. Then we shall see how it relates with meiosis. So can we have some quick answers about mitosis. Let's start by defining mitosis. What is mitosis? Yes, Nancy.
Mitosis is a process by which a single parent cell divides to produce two identical cells.
Okay, so Nancy says there's a single cell dividing to form two identical daughter cells. Is there anything she has left out that you'd want to share? Yes, Queen Esther.
Okay, for me, mitosis is the type of cell division in which a cell divides into two daughter cells, each having exactly the same number of chromosomes as the parent cell.
Okay, so she's added on that they have the same number of chromosomes as the parent cell. That is correct. That is why we say they are identical. That's why we say they are identical. If
Perhaps we want to try something.
I'm going to ask my co-host to exit those people who keep talking in the background and disrupting the lesson.
Master, can you repeat for me mitosis?
If you want to say something, you put up your hand. You don't just... Yes, Lizzie.
I was just kindly requesting if you can repeat mitosis, like the definition.
Okay. Kezabo said something. Maybe I'll ask her to repeat. What is mitosis?
Mitosis is the process whereby two daughter cells, each with the same number of chromosomes and kind as the parent cells, are formed.
Okay, so mitosis, like she has said, is the type of cell division whereby a cell divides to form two daughter cells. Okay, so the product of mitosis are two daughter cells. What is special about it? The two daughter cells are identical to each other, and they are also identical to their parent cell. So it means that they have the same number of chromosomes. The two daughter cells have the same number of chromosomes. And they also have the same number of chromosomes as their parent cell had. Okay? As their parent cell had. So that's why we are saying mitosis is the type of cell division which results into the formation of two identical daughter cells with the same number of chromosomes as the parent cell. So there are the main points that define mitosis. Now, we saw that mitosis occurs in a unique type of cells. What are those unique type of cells? There's a special name we give those cells where mitosis occurs. Starts with letter S. Yes, Mugabe.
Teacher, I'm not sure if it starts with letter S, which I know is that they're called body cells.
Okay, body cells. Another word for body cells is one which starts with letter S. We call them the somatic cells. Somatic cells means the same as body cells.
Must have not understood.
Does this topic work for the new curriculum, people?
I'm teaching people who are taking the new curriculum, and whatever I'm teaching is on the syllabus. Okay?
Yes, sir.
Yeah. But next time you put up your hand and ask, because you keep disrupting. If you keep interrupting like that, we lose valuable time when everybody wants to talk.
Okay.
Those who are annotating, please stop it. Yeah, little Pry. How can I help?
I'm requesting if you could pardon on the other name of the cells where mitosis takes place apart from body cells, the other name that you said.
Okay, I'll repeat it, but you see when people keep re-interrupting, that's what happens. So we said they are body cells. Another word are somatic. S-O-M-A-T-I-C. Somatic cells. Somatic cells. So the word somatic means body cells. Okay, it means body cells. Now, if they are body cells, it means there must be other type of cells besides body cells. What are the other types of cells? If you are very observant, you will already see the answer. Little Pry?
Master, how do
Yeah, Queen Esther.
Excuse me. I've
I think sex cells.
Yes, they are called sex cells. Another word for sex cells starts with letter G. Yes, Mary Shanik.
Sperms and egg cells.
Yeah, but want one word. That's correct, but want one word to represent sperm and egg cells. Mwebaze?
Genetic cells.
Not quite. You are almost close. Ida? Okay, Josie?
Gametes.
Yes, they are called gametes. Okay. So gametes are formed by another process that is not mitosis. They are formed by a process called meiosis. So you can see the word in red here, meiosis. It's the one which results in formation of sex cells, or what we call gametes. And once they are formed, they are important for a process known as sexual reproduction. And it involves what we call fertilization. So the male and the female gametes come together to form a zygote. And so today I want us to look at this process of meiosis and see how it takes place. So I'm going to play a video and I want you to watch it very intently and very attentively so that you do not miss out a thing. We shall discuss the video afterwards.
Sperm meets egg, which then undergoes multiple cell divisions to make a living organism with trillions of cells, all doing their own specialized function. Impressive, but where do the egg and sperm come from? The answer is, as you may have guessed, from the title meiosis. First, it's good to know that there are two types of cell division processes.
Sorry about that. I don't know what just happened. Let me see what I can do. Sorry about that. Can we see my screen? What can you see? Please read what you can see.
Inheritance.
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 Okay, that's good.
Senior for help.
Cell division.
Okay, that's enough. Let me play the video.
Sperm meets egg, which then undergoes multiple cell divisions to make a living organism with trillions of cells, all doing their own specialized function. Impressive, but where do the egg and sperm come from? The answer is, as you may have guessed, from the title meiosis. First, it's good to know that there are two types of cell division processes. The simpler one is mitosis, which produces two identical cells with exactly the same genetic information. You can think of them as clones of each other. And to find out more, why not look at our videos about mitosis? The other process is meiosis. It's a much more complicated process, creating not two, but 4 cells, with only half the number of chromosomes, and crucially, all genetically different from each other. Both mitosis and meiosis include the same phases: prophase, metaphase, anaphase, and telophase. Except in meiosis, they happen twice, so they are usually referred to as 1 and 2. The easiest way to remember these phases names is to remember IPMAT. So let's look at meiosis in more detail. As always, cellular division starts with a process called DNA replication. This involves making 2 identical copies of the original DNA molecule. The cell ends up temporarily with double the normal number of chromosomes. You can learn more about DNA replication in this video. In prophase 1, the duplicated chromosomes join up with the pair from the other parent. So the mother's pair bind with the father's pair, forming a group of 2 chromosomes called homologous chromosomes. What happens next is vital for successful meiosis, as each chromosome is lined up next to its partner's pair. One chromatid from each side gets entangled with a corresponding chromatid from the other side. This the 2 chromatids for certain sections of
So we are not able to see right now.
Okay, let me.
Teacher, we are not able to see you now.
Okay, I'm going to play it again. I don't know, the network is not so good today. Let's try again.
The network isn't stable.
Must I only sing document free coverage? Only document free coverage.
which then undergoes multiple cell divisions to make a living organism with trillions of cells, all doing their own.
It's only document recovery on the screen.
Okay, let's start this afresh because it keeps disturbing. I hope we can all see this.
The video and go on.
Can you all see the green screen?
Yes.
Okay.
Yes.
Yes.
Let's keep our fingers crossed that this time it plays out throughout up to the end.
Sperm meets egg, which then undergoes multiple cell divisions to make a living organism. Trillions of cells, all doing their own specialized function. Impressive, but where do the egg and sperm come from? The answer is, as you may have guessed, from the title meiosis. First, it's good to know that there are two types of cell division processes. The simpler one, mitosis, which produces two identical cells with exactly the same genetic information. You can think of them as clones of each other. And to find out more, why not look at our videos about mitosis? The other process is meiosis. It's a much more complicated process, creating not 2, but 4 cells, with only half the number of chromosomes, and crucially, all genetically different from each other. Both mitosis and meiosis include this.
The network is poor.
I'm very sorry, there's little I can do about that. I don't usually get this. I don't know what has happened today. But maybe what we'll do is to try one more time. Yes, Faisal?
Why don't we just leave the video, just send it to the group, let us continue studying because time is going.
I'm not sure I'll be able to share it on the group because I wanted us to use the video to be able to discuss what I want to discuss. These things are better understood when you can relate what you have seen rather than what you have imagined. So that's the challenge we have. Hello?
Teacher, me, I think it's the video with the problem because it keeps stopping on a particular part of the video.
Okay,
So I think
I can get another video. I have many of them. Maybe we can try that. I can get another video.
Excuse me, teacher.
Yes, please.
Maybe you could share the video with someone with stable network.
That may take some time. Okay, let's try to discuss without the video, although it may not make it as easy. Now, what we know, let me first close off this. Okay, what we know, can you see the inheritance video?
Yeah.
Inheritance, rather. Okay, let's just get to the notes bits.
No.
What are you seeing?
Stages.
Okay, I think we shall put our discussion on this. I'm going to illustrate using, I may draw what I need you to understand. I hope that works. Okay, so this is what we are going to do. We are going to talk about the stages of meiosis. But before we do that, let's first discuss what we had so far seen on the video. In the video, we are comparing two processes. One was mitosis, and the other one was meiosis, okay? And from the definition, there are certain differences we can clearly see. What are those differences between mitosis and meiosis? Yes, Nancy?
In the video, mitosis divides to give two identical daughter cells, and then meiosis divides to give 4 different daughter cells, not identical but different, and there are 4 than in.
Okay, thank you, Nancy. So from what Nancy has told us, the first difference is the number of daughter cells, okay? The number of daughter cells. We find that in mitosis, we have only 2 daughter cells, and in meiosis, we have only 4 daughter cells. And then there's something else she has added, that in mitosis, these daughter cells are identical genetically, which means they have the same number of chromosomes. But in meiosis, we are having non-identical daughter cells. And why are they non-identical? That one we shall find out as we get along. First of all, we said that in mitosis, the daughter cells are identical to each other, but also identical to their parent cell. In meiosis, we see that they are non-identical to each other and also non-identical to their parent cell. Okay, that is something very, very important. Is there something else that has been left out? Because in the video, there were a number of things that had so far been mentioned. Can we talk about those things? Yes, Jimmy.
Okay. When we are talking about the differences of meiosis and mitosis, we see that in mitosis, only one mother cell is required. But then in meiosis, 2 mother cells are required.
Jimmy, I think you didn't get that well. In both types of cell division, we start with one parent cell. Okay? So in both, one parent cell is involved, but it is how many daughter cells that come out of that division that makes them different. I hope that is clear. One parent cell divides, but in mitosis, it forms 2 daughter cells. In meiosis, it forms 4 daughter cells. Anything else that hasn't been mentioned? Okay. The other thing that somehow got lost as we kept playing the video back and forth is that in meiosis, the daughter cells have a half of the chromosome number of their parent. If the parent had 46 chromosomes, like in humans, the sperm will have 23 chromosomes, which is a half. The egg will also have 23 chromosomes, which is a half. And so these daughter cells that have a half of the parent's number of chromosomes, we call that state as haploid. We say they are haploid. Haploid means that the cell has half the number of chromosomes as the parent cell. Okay, that's what haploid means. So I think that was also mentioned. Okay, now let us look at the stages of meiosis, and then we shall see how the stages of meiosis are different from those of mitosis that we studied much earlier. So we saw also in the video that meiosis and mitosis both have the same names for their stages. The same names for their stages. What are the names of the stages of mitosis? What are the names? Okay, I see in the group someone has mentioned them, that there is interphase, prophase, metaphase, anaphase, and telophase. Okay, now those are the stages. Interphase is not really a stage of mitosis. It is a state when the cell is resting or the cell is not dividing. So the stages of mitosis and the stages of mitosis, usually we consider prophase, metaphase, anaphase, and telophase. Okay, although some people put interphase, but in reality, interphase is at the time when the cell is not undergoing division, so it cannot be one of those stages. Now, similarly, in meiosis rather, those stages are repeated. But there is a slight difference, that mitosis occurs once, those stages occur once. But in meiosis, those stages occur twice. Okay? Those stages occur twice. It's like if you are in senior 5, you will go through first term, second term, third term. Then in senior 6, you go through first term, second term, third term. So it's like in A level, you have 3 terms, but you do them 2 times. I don't know if that makes sense to you. For those who watch movies, you have a season, and the season has episode 1, 2, 3, 4, like that. Then the next season also has episode 1, 2, 3, 4, like that. So that is what happens with meiosis. So in meiosis, you have 2 divisions taking place. Okay, you have what we call first meiotic division, and then the second meiotic division. So the first meiotic division has 4 stages: prophase, metaphase, anaphase, and telophase. And then the second meiotic division also has the same stages. Now, how do we tell them apart? So we said for the first meiotic division, we shall call it prophase 1. Then metaphase 1, anaphase 1, and telophase 1. You get it? It's like saying I'm in S5 first term, S5 second term, S5 third term. Then when you go to senior 6, we say senior 6 term 1, senior 6 term 2, senior 6 term 3. So the phases are the same, but we separate them by adding the 1 or the 2 to know which meiotic division you are referring to. And similarly, like what happened in mitosis, you notice that in meiosis, first meiotic division, prophase 1, we start by the chromosomes condensing. Here I've not shown it, but in the video, you know that the chromosomes, when the cell is not dividing, they are very thin and not visible to the naked eye. Even under the microscope, they will not be very easily seen. But when the cell is preparing to divide, these thin thread-like structures, okay, known as the chromatin, will start to condense, okay? And when they condense, they become thick and become very visible. to the naked eye. And then they form what we call chromosomes. They form what we call chromosomes. So these chromosomes will now pair up. Remember, in a cell, the diploid cell always has maternal chromosomes and paternal chromosomes. So the paternal chromosomes are the chromosomes that came from the egg, rather the sperm that fertilized the egg and then form the zygote. Then maternal chromosomes come from the egg. So when the egg and the sperm come together, then they will form what we call a zygote. So each one of them contributes its chromosomes that it has carried with it. Okay? So when the chromosomes come together, now they are going to form what we call homologous chromosomes. It means that your mother's chromosome number 1 will look for your father's chromosome number 1, and they form a pair. Then your mother's chromosome number 10 will look for number 10 for your father's chromosome, and they also form a pair. That's what we call homologous chromosomes. So homologous chromosomes are basically chromosomes from the mother and the father pairing up in this zygote, okay, or in this cell. And we shall see why this pairing up is important. I can see some people have raised their hands. Let's listen to Jimmy first, then Tendo, and then lastly, Mugabe. Yes, Jimmy.
Like, I just wanted you to pardon the explanation of the condensing of the chromosomes, and that I didn't get it well.
Yeah, you see, that's why I wanted to play that video, okay? Because it's not that easy for you to envision it in your head, but I'll try and play that video at some point. When we talk about condensing, imagine, I want you to imagine with me, you have a piece of thread, like that which is used to make those for crocheting. I don't know if you know what crocheting is. Those things we put in the chairs to decorate them. Those vitambala, as some people call them. Now, when the thread is alone, it is thin. But when you bring so many threads together, the thing appears very thick. So that's also what happens. We have very thin chromatin, but when it condenses, it forms something which is thick, what we call the chromosomes. Let me look for the video. I think there's no other way around this. Let me try to get a video for you. Let me try to play the same video, but to that part. Maybe this one will not do. Let me get another video. Please bear with me. Let's play this one, I think, to us.
In this lesson, we'll explore the details of what happens during the phases of meiosis. Meiosis, sometimes reduction division, is a type of cell division that produces gametes.
Can you see?
By gametes, phase 1 met during meiosis 1, one from each parent. Meiosis 1.
Can you hear me?
Yes.
Yes.
On your screen?
Yes,
Yeah.
and you're still the same.
Okay, let's watch this one. I think it will work for us.
Prophase 1 starts with a
Observe
diploid.
those
Its
very thin
chromatin
things.
contains 2 uncoiled, spread out sets of chromosomes, one from each parent. After the DNA in the chromatin replicates, it condenses into the more familiar X-shaped chromosomes.
So I think you can see what has just happened. We had very tiny thread-like structures in the cell, and these structures now condensed. And when they condense, they form more visible structures, which we are calling the chromosomes. The chromosomes appear like letter X. Okay? Now, when the cell is not dividing, these chromosomes are not visible. They will go back to what we had seen at the beginning when they were thread-like and very tiny. So before a cell divides, it must first undergo this process. The chromatin must condense and form chromosomes. Okay? Yes, Cantono. Please ask your question. Okay. If there's no question, I think we can proceed. Okay, Mugabe. Mugabe, please, if you have no question, don't leave your hand up. It will confuse us. Ida.
Teacher. Teacher, so what is a diamond?
First they are small, and then
Please, when I ask you to talk, please talk quickly because there are many people who want to ask. Okay, let's have Ida first.
Okay, teacher, this is what I've understood. You're telling us they be small particles, but as they are condensing, they
Not particles, they are called chromatin. It's called chromatin.
they are chromatin.
Mm.
As they are condensing, each of them gets to find their partners to form a chromosome, right?
No, that's not what I said. What I said is this, okay? That in the cell, we have chromatin, okay? The chromatin is the thread-like structures that carry the genes from your mother and your father. Okay, maybe I not told you about that. Now, at the time of fertilization, okay, the chromosomes from your father and the chromosomes from your mother entered into this, they formed this one cell we call the zygote, okay? Are we together?
Yes.
Yeah, so every body cell has your father and your mother's chromosomes. Is that clear?
Yes.
Now, when we look at the human body, they are what we call gonads. Gonads are the special organs that produce gametes. Now, in the gonads, such as the ovary and the testis, is where this process of meiosis takes place. Now, those sperms are produced when meiosis takes place, and the eggs or ova are produced when meiosis takes place. So what happens is that as the process of meiosis is beginning, okay, or rather before it begins, each of those cells inside there has this chromatin, but it is not visible to the naked eye, okay? But if the cell is going to undergo meiosis, the chromatin now condenses, and as it condenses, it becomes visible And when it condenses, it forms what we call chromosomes, as you're seeing on the screen. Now, in our bodies, we have 46 chromosomes, 23 of which you got from your mother and 23 you got from your father. Now, during prophase, when the chromosomes have been formed, the next thing is for them to pair up. Those from your mother will pair up with those from your father. So you find that chromosomes are not of the same size. Some chromosomes are longer than others. So the ones which are identical to each other are the ones which pair up. So it's like you have the short chromosomes pairing up with short chromosomes from your father, pairing up with the short chromosomes from your mother. So now that pair of chromosomes is what we call the homologous pair of chromosomes. Homologous in the sense that they are similar. The word homo means same. Okay? So the pair of chromosomes from, rather the chromosomes from your father will pair up with those from your mother. So if you have 23 chromosomes from your father, so chromosome number 1 that you got from your father, we look for chromosome number 1 that came from your mother. Chromosome number 5, we look for chromosome number 5 from your mother. Chromosome number 6, we look for chromosome number 6 from your mother. Chromosome number 20, we look for chromosome number 20. So these chromosomes pairing up together, okay? They will form now what we call homologous pair of chromosomes. We call that process, okay, of them coming together, okay? We call it the what? Synapsis, okay? We call it what? Synapsis. And we shall see why they must come together, because when they come together, there is going to be what we call crossing over, okay? If you can just watch briefly, then you may ask the questions. For now, I'm not going to take the questions. Let's first watch. I'm sure some of the questions you have may be answered as you watch. Okay?
The replicated DNA is the same in the identical sister chromatids of each chromosome. In a process called synapsis, each chromosome pairs up with and binds to its corresponding homologous chromosome, forming a tetrad. A tetrad is the group of 4 sister chromatids in paired homologous chromosomes. The chromosomes contain genetic information called genes. These genes were inherited from each parent. And different versions of the same gene on each chromosome are called alleles. In a process called crossing over, chromatids from each homologous chromosome exchange segments of alleles. Also called recombination, crossing over randomly happens on every chromosome, resulting in different gene combinations. This explains why every gamete is genetically different from every other gamete. Crossing over results in genetic variety in offspring. This is why children are different from their biological parents, as well as from their biological siblings. Continuing on with prophase 1, the nuclear membrane disappears. The centrioles move to opposite ends of the cell, and spindle fibers fan out from them. Next, in metaphase 1, the homologous chromosomes line up at the equator and attach to spindle fibers from opposite poles. During anaphase 1, spindle fibers separate the homologous chromosomes in each tetrad and pull them to opposite poles of the cell. The cell enters telophase 1 with 1 chromosome from each homologous pair at separate poles. However, each chromosome still consists of sister chromatids. Keep in mind that each chromosome's sister chromatids are no longer identical because of the allele exchange that happened during crossing over. Then, spindle fibers disappear, and the nuclear membrane reforms around the chromosomes. Finally, cytokinesis occurs. Meiosis I ends with two genetically different haploid daughter cells. Each haploid cell contains only one set of chromosomes, consisting of paired sister chromatids. Both cells now enter the next stage, meiosis II. However, unlike meiosis I, DNA does not replicate before meiosis II begins. Once again, in prophase II, the nuclear membrane disappears, and spindle fibers fan out from the two sets of paired centrioles. During metaphase II, the chromosomes in each cell line up at the equator and attach to spindle fibers from both poles. During anaphase II, the sister chromatids of each chromosome separate and move to opposite poles. Once the sister chromatids separate, they are called chromosomes. Finally, during telophase II, the spindle fibers disappear and nuclear membranes reform. And cytokinesis occurs in both cells. Meiosis II ends with 4 genetically different haploid daughter cells, each containing only one set of chromosomes. Some key points to remember about meiosis. It begins with a diploid cell. Meiosis only produces gametes. Gametes are genetically different haploid cells, sperm cells in males and eggs in females. Meiosis has 2 stages of cell division, called meiosis I and meiosis II. During meiosis I, homologous chromosomes separate to produce 2 haploid cells, each containing chromosomes in the form of paired sister chromatids. In meiosis II, the sister chromatids separate in both cells, becoming individual chromosomes. Cytokinesis of these cells produces 4 genetically different haploid gametes. And here are some key points to remember about prophase I. The pairing of homologous chromosomes, called synapsis, occurs. Each pair of homologous chromosomes, consisting of 4 chromatids, is called a tetrad. During the process of crossing over, chromosomes in homologous pairs exchange segments of alleles. Crossing over results in genetic differences in gametes. All gametes produced by meiosis are haploid.
So I hope what you have seen has helped us get a better picture. Now I'm going to take on questions to clarify whatever misconceptions may be there. Let's start with Kelly's. Please ask your question, keep it short. Kelly's, please unmute and ask. Okay, let's have another person. Seems Kelly's has a problem. Victoria.
Good afternoon, sir.
Good afternoon.
Mine is about the diploid cell and the haploids.
Yes.
Like, I want more light on those two.
Okay, thank you, Victoria. Now what happens is this. The word diploid, if you see the first 2 letters, di means twice, okay? So haploid means half, okay? So when we look at, let me use your body. Now, your body cells, which we call the somatic cells, they are all diploid, meaning they have 46 chromosomes, okay? But your gamete cells, that means the egg cells for female and then the sperm cells for male, they are haploid, meaning they have half the number of chromosomes found in the body cells. So if your body cells have 46, then your haploid must have 23. If you look at other organisms, such as maybe the fruit fly, or you look at an elephant, or you look at a dog, they have different number of cells. So, for example, if I just say the fruit fly has, say, 8 chromosomes, okay? If a fruit fly had 8 chromosomes in its body cells, it means that its gametes would have 4. So, because the gametes must be haploid. So if an organism has 16, then the haploid must be 8. If an organism has 20, the haploid means 10. So remember that the gametes or the sex cells always have half the number of chromosomes as the body cells. And the gamete cells are produced by meiosis, but the body cells are produced by mitosis, because mitosis has to maintain the same number of chromosomes as the parent cell. But meiosis must halve them or divide them by 2. And that means if the number of chromosomes in the parent cell are divided by 2, that means then you get a haploid number of chromosomes. I hope that is clear to you, to Victoria, and any other person who had a similar question.
Yes, teacher. Thank you, sir.
You're welcome. Yes, Rosalind.
Good afternoon, teacher.
Good afternoon.
I'm requesting that you throw more light on alleles.
Okay, alleles, that is what we are supposed to discuss in our lessons that will come in the near future. Okay, but just to give you a quick one, alleles are alternative forms in which a gene may be expressed. Okay, alternative forms in which a gene may be expressed. Let me give you an example. Now, if we look at our human bodies, for instance, we all have different characteristics. Let's take the character of, say, hair, okay? Some of us have hair which is curly. Our hair is curly. Others have hair which is straight, okay? Now, the gene is for hair, but the allele, that gene can be expressed as curly hair in some people and in other people as straight hair. Or we can even, still talking about hair, we can talk about the texture of hair. The gene is for texture of hair. Some people have rough hair, others have smooth hair. So you see, smoothness of the hair and roughness of the hair are alleles for the gene for texture of hair. Still looking at hair, I can look at length of hair. Some people have short hair, others have long hair. So then we can also look at color of hair. So you can see that when you look at hair, there are many genes there are many traits by which we can describe hair. We can describe the color, the texture, the length, and so many other things. So when we talk about an allele is the alternative way in which a gene may be expressed. If we look at, say, eye color, the iris can be blue in some people, not mostly Europeans, or it can be green, it can be black, it can be brown. You get it? So the color that your iris shows is the allele for the color of eye. The gene is color of eye, but the expression can come out as blue eyes, brown eyes, green eyes, or black eyes. I hope that is clear to you, Rosalind.
Yes, teacher.
Yeah, but we shall talk about that in our next lesson, hopefully. Ida?
Okay.
Me now I'm getting confused in alleles and what.
Yeah, that's why I didn't want to talk about alleles yet. But for now, Ida, for now, You just concentrate on chromosomes. Just know that chromosomes carry genes, okay? And in your body, you have your mother's chromosomes and your father's chromosomes within the same cell. Now, at the time of gamete formation, both chromosomes cannot exist in the daughter cell. A half of them will have to enter each gamete. Are we together? So that's what we need to concentrate on for now. The alleles, I will explain them in more detail when we come to that second learning outcome, which I read to you earlier. All right, let us have another question or two. No questions? Okay, maybe I'll be the one to ask the questions. Yeah, Tendo.
So I'm requesting that you throw more light on those things of crossover that the man was explaining in the video, that thing of crossover and some other terms that he said.
All right. But did you see how it happens? Okay.
Mr. Some of us didn't see the network was not stable.
Okay, next time you put up your hand first, right?
I
Tendo.
saw, but I'm a little confused on how he explained
Okay,
it.
this is what I'm going to do. This is what I'm going to do. I'm going to play that video one more time. And you have to keep your fingers crossed that your network is very stable so that you don't miss out anything. And yeah, so let me take back the video to profess. I think let's come to profess. I think here. Ignore the alleles for now. They are not important to us. I think let's start from right here. Okay.
During meiosis 1. Prophase 1 starts with a diploid cell. Its chromatin contains two uncoiled, spread out sets of chromosomes, one from each parent. After the DNA in the chromatin replicates, it condenses into the more familiar X-shaped chromosomes. The replicated DNA is the same in the identical sister chromatids of each chromosome. In a process called synapsis, each chromosome pairs up with and binds to its corresponding homologous chromosome, forming a tetrad. A tetrad is the group of four sister chromatids in paired homologous chromosomes.
Okay, let me keep pausing, maybe to make more sense. Now, we have seen that when meiosis has just begun, we have many thread-like structures known as chromatin, some which came from your mother and others from your father. They are not visible to the naked eye, but because a cell is going to start dividing, they are going to coil and condense. So when they coil and condense, they are able now to form what we call chromosomes. Now, each chromosome is made up of two chromatids. Okay? Each chromosome is made up of two chromatids. It is like one part of the X and the other part of the X. Okay? And why are they two chromatids? Because in interphase, if you recall from mitosis, the number of chromosomes or chromatin material had to first of all double. Okay? It's like if you have some property and you want your children to have exactly the same property as you have, assuming, say, you have 4 cars, and you want each of your children to have 4 cars, maybe you have 2 children. What you must do is to buy another 4 cars so that now you have 8 cars, so that now each of them will now have 4 cars. So that's why we have the chromosomes having 2 chromatids. So the chromatids, let me just illustrate. So this is one chromatid and it's another chromatid. So these are identical because there was doubling of what was there. Like you will go and photocopy. When you make a photocopy, you get exactly what was on the original piece of paper. Okay? Yeah, so that's what the chromatids are. So we are saying that in prophase 1, the chromosomes, let's say the red ones are the paternal and the blue ones are the maternal. So the red ones, we look for the blue ones, which are of the same size, okay, which carry the same genes. So this coming together of chromosomes that are identical or are alike is what we call synapsis. Okay, so now when synapsis takes place, it means that now we have 4 chromatids instead of 2. You see this one here, and this one here, then this one and that one. So those are the 4 chromatids. That's why we call it a tetrad. Okay, so during synapsis, we have the coming together of homologous chromosomes to form what we call tetrad. Okay, so maybe we can carry on.
The chromosomes contain genetic information called genes. These genes were inherited from each parent. And different versions
ignore the alleles.
Each chromosome are called alleles.
Just ignore that for now.
In a process called crossing over, chromatids from each homologous chromosome exchange segments of allele.
So you can see what has just happened, that a portion of the chromatid, this red chromatid, has broken off and attached itself on the blue chromatid. And also a portion of the blue chromatid broke off and attached itself on the red chromatid. So that's what we call crossing over. Imagine that you have your hand. Okay, imagine you hold someone's hand as if you are greeting, and then their hand detaches and joins your arm, and then your hand also detaches and joins their arm. So it means that now you're no longer the same person because you have a hand which doesn't belong to you, like as if it has been grafted on your arm. So this is what we call crossing over. It means that the maternal genes have gone off from this maternal chromosome and attached themselves on this paternal chromosome. And the paternal genes have gone off from the paternal chromosome and have attached themselves on the maternal chromosome. So we have a crossing over. That's what crossing over is, for those who are wondering.
Also called recombination, crossing over randomly happens on every chromosome, resulting in different gene combinations. This explains why every gamete is genetically different from every other gamete. Crossing over results in genetic variety in offspring. This is why children are different from their biological parents, as well as from their biological siblings. Continuing on with prophase 1, the nuclear membrane disappears. The centrioles move to opposite ends of the cell, and spindle fibers fan out from them. Next, in metaphase 1, the homologous chromosomes line up at the equator and attach to spindle fibers from opposite poles. During anaphase 1, spindle fiber.
So you can see that in prophase 1, basically it was all about forming homologous chromosomes and then crossing over. In metaphase 1, it's about lining up at the center of the cell. In anaphase 1, it's about these homologous chromosomes being pulled away from the center towards the centrioles by the spindle fibers. Okay?
First, separate the homologous chromosomes in each tetrad and pull them to opposite poles of the cell. The cell enters telophase 1 with one chromosome from each homologous pair at separate poles. However, each chromosome still consists of sister chromatids. Keep in mind that each chromosome's sister chromatids are no longer identical because of the allele exchange that happened during crossing over. Then, spindle fibers disappear and the nuclear membrane reforms around the chromosomes. Finally, cytokinesis occurs. Meiosis 1 ends with two genetically different haploid daughter cells.
Yes, Elijah, how can I help you?
Yes, thank you so much, sir. I'm sorry to inconvenience into this meeting, but I was kind of requesting to know the difference between meiosis 1 and meiosis 2 in terms of the meiosis, cell division. I was kind of requesting to know the differences between those two areas. Thank you so much.
Okay, we shall do that. Let's first finish the video. Those who are annotating, please stop it.
Each haploid cell contains only one set of chromosomes, consisting of paired sister chromatids. Both cells now enter the next stage, meiosis 2. However, unlike meiosis 1, DNA does not replicate before meiosis 2 begins. Once again, in prophase 2, the nuclear membrane disappears, and spindle fibers fan out from the two sets of paired centrioles. During metaphase 2, the chromosomes in each cell line up at the equator and attach to spindle fibers from both poles. During anaphase 2, the sister chromatids of each chromosome separate and move to opposite poles. Once the sister chromatids separate, they are called chromosomes. Finally, during telophase 2, the spindle fibers disappear and nuclear membranes reform. And cytokinesis occurs in both cells. Meiosis 2 ends with 4 genetically different haploid daughter cells, each containing only one set of chromosomes. Some key points to remember about meiosis. It begins with a diploid cell. Meiosis only produces gametes. Gametes are genetically different haploid cells, sperm cells in males and eggs in females. Meiosis has 2 stages of cell division, called meiosis 1 and meiosis 2. During meiosis 1, homologous chromosomes separate to produce 2 haploid cells, each containing chromosomes in the form of paired sister chromatids. In meiosis 2, the sister chromatids separate in both cells, becoming individual chromosomes. Cytokinesis of these cells produces 4 genetically different haploid gametes. And here are some key points to remember about prophase 1. The pairing of homologous chromosomes, called synapsis, occurs. Each pair of homologous chromosomes, consisting of 4 chromatids, is called a tetrad. During the process of crossing over, chromosomes in homologous pairs exchange segments of alleles. Crossing over results in genetic differences in gametes. All gametes produced by meiosis are haploid.
So I hope this time a good number of us are on the same page. I'm going to deal with Elijah's question first, and we are going to deal with it together because he has asked the difference between meiosis 1 and meiosis 2. Let us deal with that question collectively. Then we shall deal with other questions that may also come up. So who wants to answer Elijah? Can we mention the differences between meiosis 1 and meiosis 2? Who can help Elijah? Please, let's stop playing videos. Those who are playing videos, please stop it. Yeah, Macrina. Macrina, please.
Thank you. In meiosis 1, during prophase, crossing over happens.
Crossing over between. You're right, but I just wanted you to give me a more complete answer.
Crossing over happens between homologous chromosomes.
Good. But in meiosis 2, prophase of meiosis 2, that's prophase 2 and prophase 1. In prophase 1, we have said crossing over occurs between homologous chromosomes. In prophase 2, there is no crossing over between the chromosomes. Any other difference, Queen Esther?
Okay, another difference I've seen is that in the first meiotic division, it forms on 2 haploid cells, while in the second one, it forms 4 haploid cells.
Yes, so the product of the first meiotic division are 2 haploid cells, and the product of the second meiotic division are 4 haploid cells. Another difference? You will notice that in prophase 1, in actually anaphase 1, homologous chromosomes are separating. But in anaphase 2, sister chromatids are being separated, if you took note. Okay, I see someone has just posted that in the chat. That is correct. Okay. What else is there as a difference? I think those are the major differences that we need to take note of. Yes, Big Daddy. Someone has a phone. Big Daddy.
The result are 2 daughter cells, each with half the number of chromosomes as the original cell.
I'm not sure I've understood what you're saying. Can you repeat, but this time while you're comparing the different types of...
In meiosis 1, the result is 2 daughter cells, each with half the number of chromosomes as the original cell.
That's not comparing. You just told us one thing. When you compare, you always have to give us the other side of the story.
Okay. While in meiosis 2, the result is 4 haploid daughter cells.
Okay. I think we mentioned that. Okay, so any other question? Should I take it that we have understood the process of meiosis? There was a summary about meiosis as the video was ending. I hope you took note of what was said there, because it's very, very important. It's very important. Okay, I think let me share again my presentation. So this video didn't work out. Let's go to the stages. These are summaries. You can see in prophase 1, the chromosomes will condense and pair up with their matching partner, what we call the homologous chromosome. And this pairing allows for genetic exchange between chromosomes, what we call crossing over. In metaphase 1, we saw that the homologous pairs of chromosomes line up randomly. The key word here is random, along the center of the cell, what we call the metaphase plate. Then anaphase, the homologous chromosomes now separate and move to opposite ends of the cell. Then in telophase, followed by cytokinesis. Now the cell divides into two daughter cells. Cytokinesis is where the cytoplasm is dividing. So we see that each of the daughter cells has a half of the original number of chromosomes, what we call haploid. Then this results, okay, I think I've just explained that. Then we go to the second meiotic division. We go to the second meiotic division. Now here we go through the same processes, more or less. We have prophase 2, then chromatin will again condense. Now at the end of the first meiotic division, that is at telophase, the chromosomes were decondensing, so they became thin and were no longer visible. So that's why here they have to condense again, okay? And when they condense again, they become now more visible. And while that is happening, the nuclear envelope breaks down to allow the chromosomes now to move throughout the cell. Remember, the chromosomes are always kept within the nucleus by the nuclear membrane or the nuclear envelope. Then in metaphase 2, those chromosomes line up along the center of each cell. Remember, now we have two cells, so in each cell, those chromosomes are lining up at the center. Then in anaphase 2, the sister chromatids of each chromosome now will separate and move to the opposite ends of the cells. Remember, whatever is happening in one cell is happening in the second cell, okay? Then we go ahead and look at telophase 2. Here we are going to see the cell dividing again. The chromosomes have arrived at the centrioles, and then now the cell divides. Now, what were two cells become now 4 daughter cells, and each of them is having the haploid number of chromosomes. Now, you will notice that in telophase, rather in meiosis 2, okay, we maintain the same number of chromosomes as at the beginning of meiosis 2. The chromosome number doesn't change. So if we started with haploid cells, then we finish with haploid cells. We don't get something lower than that. So these daughter cells are going to be genetically different from each other, as we had already seen, because there was crossing over. And again, the way they line up at the equator or at the metaphase plate was random. I want you to imagine that there's an assembly at school and you are supposed to line up according to your classes. But then now, some people line up randomly. So if they line up randomly and we come and divide the school into 2, whereby we have S1, S2, S3 on one side and then S4, 5, and 6 on the other side, we cannot have the same situation, because if some S3s go and line up with S5s, and some S2s go and line up with S6s, and some S6s come and line up in S1, then it means that we cannot have half of the school having only S1, S2, S3, and half of the other school having S4, S5, S6, because the way the people are lining up is very random. So these two things are the ones responsible for the genetic variation, the random arrangement or assortment of chromosomes, as well as the crossing over. And this is why sexual reproduction results in individuals that are different from each other. Those are siblings, and different also from their parents. Tendo's hand was up.
Teacher, I'm
Tendo.
asking, I have a question.
Yes.
Is where does the meiosis take place? Takes place in the zygote or in the human body? I'm confused.
Okay, good question. Who can help Tendo to understand where meiosis takes place? Let me see anyone in the chat. Yes, Lizzie.
To me, I think.
Please louder. We cannot hear you well. Lizzie, please.
Meiosis takes.
Okay,
I was saying that I think that meiosis takes place in the body, the body cells, not the zygote.
Not really. Jimmy? Thank you, Lizzie.
Yes, I think it takes place in the gametes.
Meiosis doesn't take place in the gametes. Meiosis results in gamete formation, but it doesn't take place in the gametes, okay? Meiosis takes place in the gamete-producing organs. What are the gamete-producing organs? Macrina?
The testes and the ovaries.
That is in animals. In plants, where would meiosis take place?
In pollen grains and the embryo sac.
In plants, it takes place in the anthers, because pollen grains are products of meiosis, okay? And takes place in the embryo sac, because in the embryo sac, that's where we have the female gametes of the plant, okay? Is that clear? Tendo's hand is still up, but before it comes up, let's listen from Sugar.
So, teacher.
Let's listen from Sugar first. Tendo, wait. Let's first give others also chance. Praise. Praise, ask your question. Okay, Praise has a problem with her device. Tendo, you may ask your question. Tendo. Okay.
I'm asking that meiosis happens like it is one that forms the sperms and what are they, ovules or what? Those things in the females.
Meiosis is the process that is responsible for gamete formation, both in males and in females.
Okay.
So the product of meiosis are gametes, and gametes are always haploid. Okay. All right. So one last thing. Okay, Queen Esther.
So my question is, in the first division of the first meiotic division, they separate and they have half the chromosomes. While in the second division, they remain with the same number of chromosomes.
Yes, that's true.
And the
Because
question is why?
yes, the second division is very similar to mitosis. Okay. For now, it may not be very easy for you to appreciate it, but I want you to know that in the second division, it is similar to mitosis. If you start with half the number of chromosomes, you end with half the number of chromosomes. That's why we say it is similar to mitosis. You understand? Okay, let's look at the significance. Let's look at the significance. Significance here, we are looking at the importance. Why is this process important in the living organism? There are three main significances, and they are as follows. One is the introduction of genetic diversity. The differences that exist amongst ourselves are because we are organisms that have come as a result of sexual reproduction. And the gametes that produced us came from meiosis. And during meiosis, we see there was crossing over, okay, during prophase 1 and independent assortment of chromosomes at metaphase 1. So these two processes are responsible for the differences between you and your siblings, your brothers and sisters. Although you have the same parents, you are different from each other because the sperm that produced them had different characteristics from the sperm that produced you. The egg that produced them had different characteristics from the egg that produced you. So you see that we are going to be different. And you may wonder, why is this so important? Now, these differences between organisms gives them what we call evolutionary advantage. It means that if, for instance, we had the plants or organisms that have certain features that make them better suited for the environment, if the environment changes, then these organisms will have a higher chance of survival. Those that don't have that characteristic will not survive. For example, if maybe one of you had a lot of hair, and then we suddenly go to winter in Uganda, okay? It means that people who have more hair may be able to keep warmer and have less chances of dying due to freezing compared to those who have very short hair. And that is coming because of your genetics, okay? So you find that some organisms are better, have higher chances of survival than others, even if they are from the same parents or they are having the same parents. Then two, we see that there was a reduction of chromosome number from diploid to haploid. You may wonder, why is it important for gametes to have haploid chromosomes? This is going to ensure that during fertilization, the offspring that is produced will have a diploid number of chromosomes. Because if that wasn't the case, imagine from the time of creation when Adam and Eve were created, it would mean that Cain and Abel would have twice the number of chromosomes as Adam and Eve had. And it means their children would also have twice. And then their children's children would also have twice. So every generation, the number of chromosomes would keep on increasing in their offspring, which would be something disastrous. But God in his wisdom decided to make sure that during gamete formation, the gametes have haploid chromosomes. So that when the gametes fuse together, then we go back to diploid. And then that organism grows, and then it starts also making its own gametes. And then the gametes are haploid. And then when that organism mates, then the two haploids join to form diploid. And the process continues like that. So you can see the intelligence that God has. Then we see finally, it is the foundation for sexual reproduction, okay? So we see that because of meiosis, we are able to form gametes. Okay, and when gametes are formed, then they are used in fertilization. And during this fertilization, there's mixture of genetic material from both parents. And that's why their offspring look like neither of their parents. They are a mixture of either parent. And that creates genetic diversity. Okay, you find that you don't resemble your mother 100%, neither do you resemble your father 100%. But you have a bit of each of them. You resemble them in one aspect or the other. And that means you are creating variety or genetic diversity, which we have seen has a big role in making you better adapted for changing environments. So I think that is all we can squeeze in the time we had. So when we meet next Tuesday, God willing, we shall look at the second learning